I want to thank Magda for giving the first talk of the fall season of this psych lecture series. As some of you know, it was started kind of in COVID, first to present some of the COVID work, but then also as a forum for people in psychiatry to learn about all the research in psychiatry. And I think it was quite successful, and there were enough people who said they'd like to see it go on, even that we've started again in the fall. It'll be less frequent, but it will have the same star cast of presenters from the department. If you'd like to present, let us know, let me know, or Rachel know. And I think for me, it's a great pleasure that Magda is giving it a kickoff for the fall season, for the 2021-2022 season. Magda received her PhD in psychiatric genetics from the Institute of Psychiatry in King's College. She went on to Oxford to do a postdoc. And then she was recruited by Avi Reichenberg to join his team as a CBER postdoctoral fellow. I think we are extraordinarily fortunate that we were able to convince her to turn down competing offers in the UK and stay at Mount Sinai, where she became an assistant professor in 2019. And in spite of all the obstacles that all of us experience, the obstacles of, coming from abroad and all that, she managed to build a very, very successful team and has really enhanced the epidemiology and related work in the CBER Center as well as in Mount Sinai. So thank you for agreeing to present, and look forward to your talk. And I'm going to unclick, but if people have questions, just use the chat function, or sorry, the Q&A function, and I will help moderate that as needed. Thank you, Magda.
Thanks, Joseph. Thanks for the very kind introduction, and thanks everyone for tuning in. I just realized yesterday actually that this will be the first time for me to present to the department as faculty. So of course it's a great honor to be here, and I'm really looking forward to sharing some of the work done in the lab with you today. And what my lab has focused on since the inception about 2 years ago is studying whether environmental causes of autism exist. And if yes, what are they? Hopefully, again, I won't lose the entire audience if I admit that we largely don't know at this point. It turned out we need a bit longer than 2 years for that. But I'm hoping to show you why it's both really important and really difficult to get this right. So I'll start from making a case for the importance of better understanding the role of the environment in autism, despite consistently very high estimates of its heritability. Then I'll discuss some of the challenges in getting things right in this field of research and how my lab has been tackling these questions for the last 2 years. And as I go along, I'll of course acknowledge the team members leading specific projects. But since these guys are such a great team, I wanted to start from giving a quick shout out to all of them. And so to give you a very brief background about what we know about the etiology of autism, thanks to some seminal studies by Mike Ratter, we've known for decades now about the prominent role of the genetic component in autism, despite at the time no insights about the specifics of, quote, what is inherited. And over the time, we've learned a little bit more about these inherited factors, as well as the genetic causes of ASD that arise de novo. So skipping over decades of discovery, really, by 2021, many specific common and rare genetic variants have been identified through the efforts of large-scale international consortia. And expanding the sample size from these 21 pairs in Folstein and Ratter study to millions of individuals linked through a range of complex familial relationships, work led by Sven Sandin from the CBER Center estimated the ASD heritability to be about 80%, adding to the firm body of evidence suggesting that ASD is of predominantly genetic origins, which exposes the huge elephant in the room, really, and makes us pose a question whether, why do we even bother studying the environment in the context of etiology of autism? and whether all the thousands of studies exploring it are either wrong or tap into a very negligible component of what is relevant. And in part, to answer these questions, I think it's helpful to spend a bit of time discussing what the heritability measures of disease actually tell us. So in brief, heritability estimate, which is this 80% in the context of autism, informs us about the variance in the phenotype that is attributable to genetic variation in a given context. So a lot to unpack, which is an incredibly useful metric, but one key caveat in inferring that genetic factors alone explain 80% of autism etiology is the non-independence between genetic and environmental effects on the phenotype. And the first issue here is the correlation between genetic and environmental variation, or in other words, the extent to which some of the environmental exposures are genetically influenced. So taking lung cancer as an example, lung cancer is always an example, we know now it can be caused by smoking, but we also know that propensity to smoke itself is under genetic influences, which already gives us a feeling that deciding whether a trait is genetic or environmental is going to be difficult. And in heritability studies, a common assumption is that trait variance is independently influenced by variation in genetic and environmental factors. But in the presence of gene-environment correlation, to estimate this effect correctly, one needs to account for this gene-environment correlation. Otherwise, if genes and environment are positively correlated, the correlation component will inflate the variance attributable to the genetic effects. And secondly, a similar inflation of heritability estimates may happen due to gene-environment interaction, which happens when certain genetic variants make us more susceptible to the effects of the environment. So under this situation, again, simplifying things a bit, neither genetic or environmental risk factor on their own are sufficient to result in disease. And the disease only occurs if both the genetic and environmental factors act together. And similarly as before, if the environmental factors tend to run in families, the variance explained by them will increase the estimated genetic effects in standard heritability models. So the heritability measures are useful to the extent they provide the upper band for the effects of genetic variation on the phenotype, but it's important to remember that they may also include the effects of gene-environment correlation and interaction. And in ASD research, we mostly still don't know how this genetic component exerts its effects on disorder risk, including potentially through the environment. And just to be very clear here, these considerations reflect a discussion in the field that really has been going on for decades. And to anyone who would like to find out more, I really recommend the Spectrum article by Brian Lee, as well as the very comprehensive review by Tanessa and Halley. But to keep the argument balanced, the other side of this coin is that the association between many of the environmental factors and ASD may occur only through background genetic confounding. So going back to this diagram, disregarding for a second everything else we know, it's plausible that smokers are more likely to get lung cancer because smoking causes lung cancer, which here is represented by the purple arrow, or because the genetic variation underlying smoking propensity also increases the risk of lung cancer, which here is represented by the navy blue arrows. But with no causal effect in that blue arrow situation between smoking itself and the disease. And it's very important to be able to distinguish between these two scenarios, because only in the smoking causal situation, decreasing the rates of smoking in the population would result in lower prevalence of lung cancer. And similar causal structure can be related to the etiology of autism with, for example, some prenatal environmental exposure and autism as an outcome. And if I was to summarize my research with a single graph, it's probably this, with all of us trying to distinguish between the purple and navy blue pathways. And to quickly illustrate this point further with one of Vache's current projects utilizing the SPARC sample, we've seen higher rates of exposures like birth hypoxia or preterm birth in children with ASD compared to their unaffected siblings, which replicates the association between these factors and autism that have been widely reported in the past. However, an interesting pattern emerged once we stratify these numbers by simplex versus multiplex family status, indicating respectively families with their single or multiple members affected with ASD. And what we saw there was for both hypoxia and preterm birth was that these exposures were considerably more common in multiplex families. And while we've only recently begun to explore what these patterns may mean, the simplest explanation for higher rates in multiplex families is that these families have higher genetic liability for autism, which accounts for the multiple members affected with the disorder, and that there is a pleiotropic effect of this genetic variation on both ASD and the exposure. And here the prevalence of preterm birth in multiplex controls, equaling the one observed in simplex cases, suggests that perhaps there are no causal effects of these exposures themselves on autism. Of course, like nothing ever is this simple, and these factors do need to be investigated further, but it's definitely a possibility we need to consider. And on the other hand, what emerged from Vache's analysis was that there were certain exposures with prevalences higher in ASD cases compared to their unaffected siblings, but with the rates in the simplex families on par with those observed in multiplex families, suggesting that familial genetic liability to ASD may be unrelated to the risk of these exposures, and the association between these exposures and autism would not be driven by genetic pleiotropy alone. These are still very early results, and we're still checking if these patterns were not just something artifactual, but the only point I want to make for now is that the relationship between our genes, environment, and autism risk are complex or may be complex, and we probably shouldn't either hype or discard things too early. And well, again, these things can be very complex, and we may not yet know what this 80% heritability estimate means. I just want to be clear that it's difficult to dispute that there is a subset of individuals with ASD whose diagnosis is attributable to the genetic causes alone. And it's fair to say that if someone has a pathogenic mutation in one of the key synaptic scaffolding proteins, likely any additional contribution of the environment is going to be minuscule or zero. And secondly, the fact that these heritability estimates may underestimate the role of the environment, it doesn't mean that they do. And as I mentioned earlier, the 80% heritability of autism represents the upper bound of this effect, but it's also possible that 80% is what it is. But leaving this complexity aside for just a very brief moment, environmental research on ASD is a really big field, and as illustrated here by a graph from the meta-analysis by Dr. Magdalena, also from Sinai, highlighting the range of environmental exposures associated with ASD in previous studies. And the factors significant in the meta-analysis included a bunch of things: prenatal exposure to SSRIs, birth trauma, maternal gestational diabetes, obesity, cesarean delivery. And importantly, over and over again, and perhaps I think now it's time to say it again, vaccines were not to any extent associated with autism risk. But as you see, there are these really long bars along the y-axis, indicating that there remains a huge amount of uncertainty with regard to the strength of the association between these exposures on ASD, which may reflect variable exposure definitions across the studies and/or estimates obtained from relatively small samples. So for example, for birth trauma, this effect is anywhere between 50% higher risk to 16-fold higher risk, which are quite different numbers, if you ask me. But except for these statistical or measurement issues, as I've signaled before, many of the epidemiological studies do not account for the potential genetic confounding I've touched upon in one of the earlier slides. So for example, an association between maternal medication use and ASD can be observed even if the true causal association between two is absent. But instead, as in a classical confounding scenario, there are shared genetic factors pleiotropically affecting both the disease risk for which the medication is taken and ASD. And the second caveat here is that the environment is more complex, and we do not really get to study these exposures like cesarean delivery in isolation from everything else. And to illustrate the point about this complexity, I'll focus on SSRIs, which is a class of antidepressant medications that have been consistently linked with an increased risk of autism, leading to a debate of the role of potential confounding by indication, i.e., whether the observed associations are not predominantly driven by the effects of maternal depression or even its underlying genetics. So ours and other groups have done some work in that direction, suggesting that this confounding is likely the case. But to complicate things even further, it also seems that depression itself is not an isolated risk factor. So this graph is taken from another project that Vahe Kachaturian has done in the lab, and it's now also available on medRxiv. And it illustrates quite well that women with a mental health disorder in pregnancy, which included mostly women with depression and anxiety, have higher rates of a series of physical conditions, including digestive, circulatory, and endocrine conditions, which really highlights the possibility that the effects of depression or its underlying genetic factors on autism risk may be observed partly due to the correlated effects of these additional physical comorbidities in mother during pregnancy. And this likely applies to a wide range of environmental exposures that we often think of and try to study in isolation. Some exposures will occur in clusters we're not always aware of. And while it's difficult to always account for everything analytically, our interpretations of the observational associations should always acknowledge this complexity. So I think it will take really intense efforts to move beyond these observational associations to a real understanding of why are we seeing these effects. But I do think that it's something that is worth our while, at the very least to conclusively show that some of these exposures do not cause autism and lessen potential fear around them. Secondly, if all we ever get to will be showing that all environmental associations we see arose due to background confounding alone, knowing about the pleiotropic effects of some genetic factors on both, for example, depression or preterm birth and ASD, is still important in understanding the etiology of autism. So having justified myself for the last 15 minutes or so, for the remainder of the talk, I'll focus on several studies done by the lab members. So this will be mostly short vignettes to highlight some of the ongoing projects, but I'll be happy to elaborate on the details later on during the Q&A. And just as a short primer, our lab's focus is on early life risk factors for autism, particularly those that can affect the fetus before birth, in line with the early neurodevelopmental origins of autism. And while we're interested in the associations themselves, our focus really is on distinguishing the potentially causal associations from those that arise due to various sources of background confounding, including by factors like parental, genetic, medical, or demographic factors. So the first study I'll highlight focuses on the role of maternal health in pregnancy in etiology of autism. And while several associations between maternal diagnosis and offspring have been suggested, including maternal depression or asthma, lack of any systematic studies means that majority of maternal diagnoses have never been looked at in the context of etiology of ASD. And also for the previously reported associations, we mostly do not know to what extent they are driven by genetic confounding or other maternal comorbidities. And addressing these knowledge gaps is important to do to better understand the etiology of ASD, to at least give ourselves a chance to identify potential preventive strategies, and to improve the design of other epidemiological studies by understanding which maternal health factors could act as potential confounders. And this project gave rise to the first funded R01 application in the lab, where our aims are to identify maternal diagnosis in pregnancy associated with the risk of autism in offspring, using both maternal medical records from Danish register and Kaiser Permanente Northern California cohort. And secondly, to interrogate the level to which these associations are driven by genetic confounding by triangulating both family and genetic data. So in a pilot study to get preliminary data for this proposal, we ran this diagnosis-wide association study between maternal conditions in pregnancy and offspring risk of autism. And this graph represents the significance levels of individual ICD diagnoses, which are color-coded by their broader diagnostic categories. So the higher the dot, the more significant the association. The gray dots at the bottom indicate diagnoses that were not significant after a rigorous filtering process, including adjustment for confounders, multiple testing correction, and permutation testing. And diagnoses that remained significant after all these steps were then analyzed in a joint model in order to account for the comorbidity between them. And we ended up observing that some genitourinary, metabolic, digestive, and psychiatric diagnoses were independently associated with an increased risk of autism in offspring. What was more curious, though, so to say, was that there was also a range of maternal diagnoses associated with lower risk of offspring ASD. And this list mostly included either mild diagnoses or medical codes indicating contact with a healthcare provider. And so it seems that likely what is driving these patterns is that women with higher healthcare utilization and therefore more likely to receive relatively mild diagnoses are also less likely to have a child with ASD. We're still thinking of some explanations as to why this could be the case, including lower subclinical mental health problems in women with higher healthcare utilization, but we still haven't really produced an explanation we feel satisfied with. But the key message here, I think, is that these associations should not be ever taken at face value. And I think there is something interesting going on here, and definitely something we need to think more about, but it's definitely not that tonsillitis or dermatitis cause autism. And since the Israeli study under, again, star postdoc Vahe Kachadurian's lead, we've refined our approach a bit and moved to a much larger Danish sample. So the preliminary findings from there to an extent replicate those in Israel, with some differences in the specific diagnosis associated with ASD, but falling into the same broad categories, with the exception of the genitourinary ones, which were not significant after comorbidity adjustment. Two additional clusters of diagnosis that emerged included injuries and birth complications. And similarly as in Israel, we observed this pattern of mild diagnosis and codes indicative of contact with healthcare associated with lower risk of ASD. After this stage, we separated non-chronic conditions and explored the evidence for the underlying genetic confounding using comparisons between exposed and unexposed siblings, which allowed us to control for family-level confounders. And also we compared the effects of maternal diagnosis occurring in pregnancy to those that are more temporally distal from pregnancy. These two analyses can't really be done for the chronic conditions because the timing of the diagnosis in the register reflects when the woman saw the doctor for her condition rather than the disease onset. And because these events, seeing a doctor and disease onset, may really be years apart, would really have very low confidence in deciding which SIB was exposed and which wasn't, and when relative to pregnancy the disease really started. But for the results, first for the sibling comparisons, what we found using a stratified Cox regression was that there was no evidence for family level confounding. And similarly, these non-chronic conditions, when diagnosed 2 to 4 years before pregnancy, were mostly not associated with autism risk, again indicating lack of evidence for familial confounding. So while we haven't identified any evidence for genetic confounding in these associations so far, we're really at the very start of this project, so things may still turn around drastically. And over the next 4 years, we'll be working to replicate these results in a Californian sample, to incorporate additional designs for investigating the genetic and non-genetic effects underlying these associations, which will include paternal and cousin comparisons, as well as polygenic risk score approaches. And if we still at that point see that there's no evidence for genetic confounding, there's plenty to be done in terms of studying where the non-genetic signal is coming from. So here by design, we're not able to distinguish between factors like direct diagnosis-related effects, maternal genetic variation that is not transmitted to offspring, but it can influence still some other ASD-relevant factors, maternal use of medication, and likely many other factors. And the second quick vignette I wanted to show is the work on the role of maternal exposure to medication in autism. So we're definitely not the first ones to think about this, with multiple studies having already shown that several classes of medications taken by pregnant women, which included antidepressant SSRIs, antiepileptics, and asthma medicines, are associated with an increased risk of ASD in her offspring. And if the medication used in pregnancy is causally related to ASD, it's of course critical that we know it. But the problem in this field is all the background confounding going on, including by maternal indication. And as I already mentioned, it's really difficult to get around it. But like some of the reasons why it's important to get this right is, first of all, some medications may be mechanistically associated with ASD. And secondly, because right now, many women may stop taking the medication they need, only because it was shown to be associated with autism due to background confounding. And so I think there is a lot of responsibility in how we do and how we communicate this research to others. And our approach to study this effect, while reducing the confounding by the underlying diagnosis, is to create these groups of medications based on their pharmacological target. So as you see here, JAKs A to E are prescribed for different conditions, as indicated by differently colored dots, but they all exert their effects for acting on the same target. And once we get this target-based group of medications, composed of medications prescribed for diverse conditions, we can test whether maternal exposure to its underlying JAKs is associated with offspring ASD. And to the extent that we do get such indication heterogeneous group, this approach reduces the confounding by indication by loosening the tight link between the JAK use and condition present in traditional approaches. It also allows us to increase power to identify true signal, because there will be more children exposed to the target group compared to the individual medication. And finally, by teasing out the mechanistic signal, it can also highlight the biological systems that are sensitive to prenatal disruption by maternal medications. This signal is also easier to replicate in other cohorts, because the medications underlying each group may differ without any consequences on the inference about the mechanisms. And this is a project that we've really been working on for a number of years in collaboration with the Department of Pharmacological Sciences which really has been one of the most fruitful and fun collaborations I've experienced. And Vahe, who is spearheading this analysis, has some really exciting updates, which you will get to find out about soon. And in the meantime, if you're attending the ACNP this year, I really encourage you to check out his poster there. So maybe a bit of suspense in terms of the latest results, but I do promise it will be worth it. And with the next vignette, I wanted to illustrate how studying the environment in autism may help us better understand not only what are the factors associated with the disorder, as in binary status of either having ASD or not, but also those factors that contribute to its differential presentation in different individuals. And this time I'll actually show you some hot off the press results from the recent project using the Spark sample. So here we focused on additional comorbidities of ASD, which, just as a quick background, are known to be highly prevalent and really affect all body systems, including epilepsy, gastrointestinal symptoms, or ADHD. And what's important here is that different individuals would present with a different constellation of these. And what we still don't fully understand is what drives the association of these conditions with autism and why different individuals can present so differently from each other. And part of the rationale for the design of this study, which is currently being finalized by Vahe, were the observations from syndromic forms of autism caused by, for example, mutations in ADNP or SHANK3 genes. And both of these mutations are associated with both ASD and a constellation of other medical issues. And so what we wanted to ask was whether, in a similar fashion, environmental factors associated with ASD are also associated with different constellation of additional comorbidities. Furthermore, we wanted to test whether there is any evidence that these comorbidities arise as a downstream consequence of autism or are causally independent of it. And before I move any further, nothing we did here implies that we believe that any of these associations between the environmental factors and autism comorbidities is truly causal. So in all our conclusions, we account for the fact that what may really be going on is that these exposures simply tag underlying causal genetic factors. So to start unpacking all of this slowly, first we did observe that all of the environmental exposures that we wanted to study occurred at a higher rate in children with autism compared to their unaffected sibling, illustrating first the association between these factors and autism, potentially confounded by the background genetics. So we've got this part of the model already, and next we observed that among autism cases, these different environmental risk factors were associated with different comorbidities. And what struck us here was that among the strongest associations we observed, many of them were already established in the general population, including, for example, association between early brain trauma and epilepsy, antenatal infection and deafness, lead poisoning and obesity, or fetal alcohol syndrome with microcephaly. And this really underscores that the mechanisms underlying comorbidities in autism may not be qualitatively different from what we see in unaffected individuals. And to explore this idea further, we looked at the association between the same exposures and comorbidities in unaffected siblings. And by doing so, testing whether the association between the environmental factor and these comorbid conditions are somehow mediated by autism. And here, because of the sample size for the siblings in the Spark study, the sample size, there were many fewer unaffected siblings compared to autism cases in the Spark study. Therefore, we focus on the two most common exposures, including preterm birth and birth hypoxia. And what we observed for both of them was that while the point estimates were often actually higher in siblings, the confidence intervals around these effects were very much overlapping in cases and controls, suggesting that there is no evidence that the association between these environmental factors and comorbidities has anything to do with having or not having autism. And so for the interim summary, it does seem like certain environmental exposures are associated with both ASD and its comorbid conditions, with no evidence that ASD is a mediating factor. And for now, the way we interpret this result is that first, autism and these comorbidities share some of their causes. And secondly, higher prevalence of these comorbidities in autism may simply be a dosage effect, with cases having more of the underlying cause, be it environmental or genetic. And now for the next penultimate vignette, I want to change gears a bit and give you a quick primer on some of the epigenetic work happening in the lab. So here, the rationale behind this work is that to some degrees, both genetic and environmental factors may influence the risk of autism, and because they both affect the epigenome, looking at things at this level, we may be able to capture both of these sources of effect. And focusing on DNA methylation, similarly as the genetic contributions to autism, we expect some effects to arise due to both common and rare or private events. And for the common side of things, we've got a well-established classical EWAS approach, which is largely analogous to GWAS. And the Manhattan plot I'm showing over here is coming from the latest published ASD EWAS study, illustrating quite well that we're probably still underpowered to detect common epigenetic signal in a heterogeneous disorder like autism. For the rare epigenetic variation, methods are much more recent, and there is still no gold standard in the field really. But Andrew Sharp's team at Mount Sinai has done amazing work at advancing this field, and the results shown in the next couple of slides rely strongly on their work described by Barbosa et al. So when it comes to these rare DNA methylation outliers, or these rare to private events, most of these events are only seen in 1 or 2 individuals, not allowing us to assess the penetance and association with ASD of any of these individual events. However, what we can do instead is do a simple burden test, checking whether these events are more common in any of the diagnoses which we're interested in, which in our study included autism, ADHD, comorbid ASD, ASD, ADHD, etc. And we ran this analysis in 2 independent Danish array-based samples, iPsych Minerva and iPsych, both of which profiled DNA methylation using neonatal blood spots. And the results from these 2 samples were subsequently meta-analyzed together. And this work, as shown here, was supported by a great collaboration with the iPsych Consortium, as well as my NARSET grant from the BBRF. And we observed slightly elevated burden of these DNA methylation outliers in both ASD and ADHD, but only in the comorbid cases, these effects were statistically significant. And this really links well with observations of higher clinical severity and genetic burden in these comorbid individuals. We also observed in both samples an increased load of these rare epigenetic events in individuals with intellectual disability. And given that intellectual disability is enriched for these rare genetic mutations, these results tie in with work done by Michael Breen and Sylvia de Rubens from the Seaver Center on epigenetic disruption in some carriers of rare genetic variants. Next, given our interest in the early environment, we tested whether certain early life environmental factors are associated with higher burden of these rare epigenetic variants. And hopefully by now it's clear that when I say environmental, what I refer to is measured on the environmental level, acknowledging the possible background genetic influences. And among these environmental exposures, We observed an association between the burden of these rare DNA methylation variants and measures of prematurity, both measured in weeks and grams and inferred from the DNA methylation data itself. So there is definitely some interesting signal to follow up on. And right now we've got another super talented postdoc in the team, Dr. Christine Hansen, on the task. And over the coming months, she'll be using the sample of mono and dizygotic twins, who are both dis and concordant for mental illness and family trios, to detect this rare epigenetic variant and further interrogate the heritability of these events, to strengthen the evidence for their potential pathogenicity, assess the recurrence across different psychiatric disorder, and last but very much not least, she will contribute to the methods development in the field. So watch the space and look out for Christine's work. And in fact, you may do so very soon if you're attending either WCPG or ASHG, where Christine will be contributing an oral and a poster presentation. And last, but very much not least, just wanted to give you a quick primer on what Artemis has been busy with since joining the lab literally a few weeks ago, which is trying to better understand the potential causal role of preterm birth in autism. And while the association has been extensively reported, including by the recent study by our wonderful colleague Sven Sandin from the Seaver Center, it's possible that despite careful controlling for the background confounding, including, for example, maternal health or smoking in pregnancy, some residual confounding remains. And of course, we can never be sure that the reported associations are truly causal, but seeing that the results from several different methods all point in the same direction, we may start feeling a bit more confident about what we're observing. And one such additional alternative approach that we can add to this battery of tests is Mendelian randomization, which, simplifying things a bit or a lot, allows us to test whether individuals who genetically are at a higher risk of an exposure are also at a higher risk of the outcome. And what Artemis is currently working on is meta-analyzing the existing gestational age GWAS data to identify genetic loci that are robustly associated with gestational age, which will serve as genetic instrument in this model. And in the next stage, this will also allow her to test whether the association between pre and post-term birth and autism occurs through the same or distinct mechanisms. And finally, in the near future, our goal will be to interrogate whether changes in DNA methylation, which we know is strongly associated with gestational age, mediate any of the effects of gestational age on either autism or other outcomes associated with pre or post-term birth. So for the last time today, do look out for the updates by the team members. And yeah, I think that's it in terms of the results. And in conclusion, I hope to have managed to convey that the environment is worth studying. And if all we ever achieve is proving its lack of involvement in autism etiology, I consider that an effort that is worth our while. The high heritability estimates may indeed reflect the fact that autism is predominantly genetic in origins, but given the impact of gene-environment correlations and interactions, I think it's good to keep an open mind about this for now, at least. And again, there are some autism cases with fully genetic etiology. We just don't know yet what fraction of all ASD cases they really are. Confounding is pervasive, but with a mix of observational, genetic, and epigenetic data, I think we can slowly work our way through this. And some of the examples of how we try to approach this in the lab include projects on maternal health and medications in pregnancy, for now trying to distill respectively familial and clinical confounding. And importantly, even if these things are still a bit murky, even without fully understanding the causal relationships between different factors, studying environment in autism can still help us better understand the underlying architecture of risk and disorder heterogeneity. And last but not least, integrating epigenetic and Mendelian randomization insights may help us better understand the functional aspect linking these exposures to neurodevelopmental and neuropsychiatric illness. And the lab is currently doing both applied and methods development work in this direction. And with that, I'd like to thank all of the amazing collaborators who contributed to different projects that were part of this talk, both within and outside Sinai. First and foremost, thanks to my amazing team and Avi and Joe from the Seaver Center. Avi for mentoring me for the last 10 years since I started my PhD, and Joseph for his help in joining and navigating early years as faculty here. And I'd like to thank many of the wonderful faculty mentors here at Sinai, my funders, and all of you for listening. And I'm ready to take questions if there are any.
Magda, let me change the view. So great talk, and to remind people, questions should go in the Q&A. Maybe I'll kick it off. So when I try and disk genetics, which I do sometimes, but rarely, and especially heritability estimates, you know, the heritability of height is even higher than the heritability of autism.
Yeah.
But height has changed systematically over decades, right?
Mm-hmm.
And so clearly there's an environmental component that's not measured. And the reason it's not measured is because there's a huge cohort effect, right? And people look at a certain cohort. And so obviously the diagnosis of autism has changed dramatically, even more dramatically, I would say, than height. So I guess I should know this, like how in the Sandin paper, how is that dealt with? And is there a way to incorporate that even with Mendelian randomization, right? Because the diagnosis, I'll shut up.
Yeah, I think, so first answer is that I don't know really whether and how that's handled in Sven's paper, in Sandin et al. But I think that exposes one of the key things about heritability estimates that maybe are not discussed often enough. But heritability estimates assume they are very specific to given context. So if, for example, we were to completely equalize everyone's environment, then all the variance you would see would be completely genetic. Heritability would be 100%. If we were,
if everybody smokes, lung cancer would be fully genetic. Is that
exactly? That's So the way we react to this environment is, determined by genetics, and that's what we would see in heritability estimates. If, on the other hand, we were to give, let's say, force all women to undergo, let's say, assuming things are causal, undergo, for example, birth complication or take some teratogenic medication in pregnancy, same, we would see higher effect of genes over environment. So I think both within certain period of time, between different cohorts, as well as within the same, let's say, site over the time, we will see these estimates change, which exposes this artifactual thing about however we call it, about heritability estimates, that they are not something that's immutable characteristic of the disease itself, or trait, as you kind of gave an example for height. And I think maybe it's often not spoken enough. I think it comes up sometimes in discussions about educational attainment and trying to estimate heritability of education or socioeconomic status. The heritability may be lower because schooling is universal, but once we take away universal schooling, heritability estimate would go up because we would see this effect.
There needs to be variance to measure the variance.
Exactly.
Yeah.
That's a short answer.
Yeah, and I guess what I often say in this circumstances, because of all the reasons you said, I honestly don't know if it's worthwhile arguing whether heritability of a certain disorder or trait is 60, 50, 70, or 90. I want to know if it's non-zero.
Yeah. Yeah, exactly. Yeah. And I used to, like, in the center where I did my PhD, like estimating heritability was one of the things many people did for many years. And I remember one of the academic being like, oh, it's always between 40 and 80. Like, what does it matter?
Well, it's good to know that it's non-zero if you're going to change it.
Yeah, I guess so. No, I think, like, no, I think I don't want to be like, I'm not at all critical about the measure. I think it's a very good measure once we understand exactly the caveats around it.
Yeah. I don't see people, maybe they're using the chat. People are use the Q&A. I will also check the chat, but please, I know you have questions out there. Oh, give it one more minute. So, happy to have open-ended discussions about anything. So, one thing that maybe you could expand upon is obviously epigenetics and environment and genetics. You know, they obviously intersect, right?
Sorry?
They somewhat intersect, right? But particularly the idea that environment is somehow reflected in the epigenome. I know that's been a long-term interest of yours. Like, where are you going with that?
Where are we going with that? I think for now we're as I highlighted, there are two main avenues we're trying to take. One is through Christine's project, looking at the rare events and to what extent they may actually contribute to etiology of ASD, to us better understanding impact of, even like understanding impact of genetic influences on risk. And secondly, I think Project Artemis will move on to, which is looking in integrating DNA methylation measures with Mendelian randomization concept framework to better understand to what effect, to what extent these things can actually mediate the effects of exposure on outcome.
Yeah,
I think we're still very limited by the sample sizes that are available.
Yep.
That's the critical thing. And, you know, for what's available, often these samples are not very well characterized. Yep. So I think, like, at least for now, a lot of work done in this sphere remains preliminary.
Yeah. So there's a related question from Michael about DNA methylation. Can you elaborate on some of your analytical approaches to study epigenetics in autism, i.e. predictions about epigenetic age or genotype-based predictions of DNA methylation, which you kind of just touched upon, but take it away.
Yeah, I'm trying to unpack Michael's questions.
Epigenetic age, and then kind of editing,
yeah.
like methylation QTLs, which I guess would roll into your Mendelian
randomization. I think both are kind of widely studied in the context of psychiatry. These are not really like things we incorporate into our analysis. Things we often account for. But yeah, we don't really explicitly study these things. And everything on paternal side of things. I was asking
about the other thing. You talk about A, and they want to know about the complement of A. So Avi wants you to talk about paternal.
I spent my PhD studying paternal, and he knows that. I think that's
the paternal side of things from Avi.
Yeah. So because men do not carry pregnancies, I think there is like one critical kind of period of exposure is out of the equation. Having said that, you know, the whole field of better understanding the effects of environmental on sperm, on offspring is very active. I think because of how many kind of connections we need to make, I don't think we're at the point where we can say any of the paternal exposures are causal of offspring outcomes through the effects on the sperm. But that's, of course, like a subject of active investigation. And yeah, often the way we're using that is as a negative control for exposure in pregnancy. That may be, given the effects on the sperm, that may be too simplistic. But yeah, something we're actively thinking about. And of course, paternal age, which my PhD was on, and Avi paid for the mice. My paternal age is like probably the most well-established risk environmental. It's complex, but risk factor for autism.
So maybe to go back to Michael's question. So if you're thinking about, you know, if you have a good kind of GWAS for autism,
yeah,
could use, you know, a kind of methylation QTLs in your Mendelian randomization.
Yeah, and I think that's how they are actually used in the framework. That's what's kind of you need for the method to work. You need the background kind of genetic loci underlying the CPG loci you're interested in. So the MQTL, sorry, I missed that. MQTLs are actively like part of the framework. I can forward the relevant paper, Michael, later on.
Okay, well, thank you for a great talk, and happy fall to everybody, and happy one of the many new years started recently. Magda, thanks for a great talk, and
and thanks everyone.
Launching this series again.
Thanks everyone.
Yeah, thank you everybody.